So I want you to imagine like a microscopic Trojan horse.
Okay.
It escapes the protective walls of your gut, surfs through your bloodstream, and then purposefully seeks out a tumor.
Right.
But it doesn't attack the tumor. Instead, it detonates. It releases this hidden viral payload that literally brainwashes your own cells into building a localized stress hormone factory.
It's wild.
And it does all of this just to protect the cancer. So today, we aren't just talking about that old cliche, that stress is bad for you.
Yeah, we've all heard that.
Right. We are looking at the exact terrifying biological chain reaction that actually proves it.
It really is, one of the most remarkable discoveries in recent oncology. For decades, the medical field has recognized that psychological stress accelerates tumor growth.
Sure.
We knew the mind and the body were connected, but the actual physical bridge between a feeling of anxiety and the rapid division of a cancer cell, that was basically invisible.
We just saw the beginning and the end.
Exactly. We were looking at the start of a road and the end of a road with, absolutely no idea how they connected in the middle.
Well, and now we have the map. So welcome to this custom-tailored deep dive. We are unpacking a truly staggering 2026 paper published in the journal Cancer Cell.
Bashir and colleagues, yeah.
Right. And they didn't just find a link. They mapped out this crazy multi-step biological sabotage circuit. It involves your stress levels, your gut microbiome, hidden viruses, and immune paralysis.
It's a localized attack happening right inside the tumor.
Orchestrated by players you would never, ever expect to be working together. Okay, let's unpack this.
What's fundamentally paradigm shifting here, I think, is the localization. When we think of stress, we usually think of a systemic, body-wide response.
Like your heart racing, your palms sweating.
Right, exactly. But Bashir's team uncovered a highly targeted microclimate of stress. It's built on demand, specifically to cripple the immune system exactly where it hurts the most.
So let's trace this from the very beginning. To understand how a tumor exploits stress, we have to start with how the body experiences it.
Yeah, let's start with the mice.
Right. The researchers use something called the CUMS model on mice, which, as I understand it, stands for chronic, unpredictable, mild stress.
That's right. And the key words there really are unpredictable and mild.
So not a massive trauma.
No, not at all. They aren't simulating a predator attack. They are simulating the relentless, low-grade friction of just daily life. So for 21 days, these mice were subjected to this rotating wheel of minor annoyances.
So we were talking about things like briefly tilting their cage at an angle, right?
Yeah, or leaving a bright light on overnight.
Or playing random white noise. It's the biological equivalent of like your car breaking down, followed by a leaky roof, followed by your alarm clock failing to go off.
Exactly the kind of week that just leaves you totally exhausted.
Right. And the researchers found that by day 7 of this protocol, the mice experienced a massive spike in a hormone called corticosterone.
Corticosterone is it's the primary stress hormone in rodents. It plays the exact same role that cortisol plays in humans.
Okay, cortisol, we know that one.
Yeah. So your brain registers the unpredictability, signals the adrenal glands, and they just flood the bloodstream with this hormone to keep the body on high alert.
Makes sense.
But Bashir's team tracked where that hormone was actually docking. They followed it straight down into the gastrointestinal tract, where it bound to a very specific receptor on the intestinal walls called NR3C1.
Okay, NR3C1. Let's pause and visualize this because I love a good analogy.
Go for it.
Think of a receptor like a highly specific keyhole on the surface of a cell.
Right.
The stress hormone is the key. When corticosterone floods the gut, it inserts itself into these NR3C1 keyholes. And this is where I think of like a castle under siege.
I like that. The gut barrier is the fortress wall.
Exactly. Its sole purpose is to keep the trillions of bacteria digesting your food safely outside of your sterile bloodstream. But when that stress key turns in the lock, it essentially signals the fortress to dismantle itself.
And the mechanics of that dismantling are surprisingly thorough, like really thorough.
How so?
Well, the fortress wall is held together by cellular mortar, what we call tight junction proteins.
Okay, the mortar holding the bricks.
Right. Specific ones like OCON and KLDN1, they literally bind the cells tightly against one another.
OCON and KLDN1. Got it.
So the stress hormone signal downregulates the production of these proteins. It stops making the mortar, and the existing mortar just dissolves.
Wow. But it doesn't just stop at the mortar, does it? It actively targets the castle staff, right?
It does, yeah. The intestinal lining replaces itself rapidly, relying on stem cells, specifically LGR5 stem cells.
So those are the masons rebuilding the wall.
Exactly, the LGR5 masons. And it also relies on immune defense cells called Paneth cells, specifically lyse-1 positive ones.
And those are the guards on the wall.
Right, keeping the bacteria at a safe distance. But the corticosterone binding to that NR3C1 receptor actively suppresses both the masons and the guards.
So your body just stops repairing the wall, fires the entire security team, and melts the concrete, holding the bricks together.
That is exactly what happens.
And to prove this wasn't just, a theory on paper, the researchers ran this fascinating dye test.
Oh, yeah, the FITC dextrin.
Right. They fed the mice a fluorescent tracer called FITC dextrin. In a healthy, unstressed mouse, that dye stays inside the digestive tract and just eventually passes through.
But in these stressed mice...
The dye leaks straight through the dismantled gut walls and lit up their bloodstream under a microscope.
It is the literal physical manifestation of psychological stress. The barrier just becomes highly permeable.
Okay, so let's do a quick recap here to lock this in.
Sounds good.
Bullet 1, psychological stress spikes your corticosterone levels.
Yep.
Bullet 2, that hormone hits the NR3C1 receptors in your gut.
Right.
And bullet 3, that signal kills the guard cells, melts the tight junction mortar, and leaves you with a leaky gut. So a question for you listening right now. Have you ever noticed your digestion going completely haywire during an incredibly stressful week at work?
I know I have.
Right. What we are looking at here is the microscopic blueprint of that exact feeling. Your psychological stress is chemically commanding your gut to drop its defenses.
And because those defenses drop, something gets out.
Right. The fortress is breached.
The breach allows the contents of the gut to spill into the blood. But, and this is key, this isn't just a random stampede of bacteria.
It's someone specific.
Yes. The researchers isolated a very specific bacterium that seemed uniquely positioned to take advantage of this stress-induced leak. It's called Enterococcus gallinarum, or just egg for short.
Okay, egg.
Yeah.
But wait, I buy that the gut gets leaky. I've heard of leaky gut. But the bloodstream is a highly policed environment.
Oh, absolutely.
It's packed with white blood cells whose entire job is to hunt down and annihilate stray gut bacteria. How does this specific egg bacteria not get completely shredded the second it enters the blood?
That was the crucial question they had to answer. When they analyzed the egg bacteria that successfully made it out of the gut and all the way to the tumor, they found it wasn't the standard run-of-the-mill gut flora.
It was different.
It was a highly adapted, mutated variant. They actually distinguished between the normal egg gut and this new egg tum, the tumor colonizer.
So it evolved specifically for this journey. What kind of armor was it wearing?
Well, it had upregulated mutations in specific survival and virulence genes like YHMG.
YHMG.
Right. Those helped it withstand the immune system's initial attacks in the blood. But even more interesting was a mutation in a gene called CHER.
CHER. C-H-E-R. What does that one do?
CHER acts like a chemical GPS for bacteria.
Oh, wow.
Yeah, it allows them to sense chemical gradients in their environment and literally swim toward favorable conditions.
And what is more favorable to a mutated rogue bacteria than a tumor?
Exactly.
I mean, tumors are notoriously chaotic. They are highly acidic, incredibly low in oxygen hypoxic, right?
Totally hypoxic.
And full of dead tissue. For a human cell, it's a total nightmare. But for a mutated bacteria with a chemical GPS, it's prime real estate.
It's a sanctuary. The HAL mutation allowed the egg bacteria to essentially sniff out the hypoxic, low-oxygen environment of the tumor, navigate through the bloodstream, and embed itself directly inside the tumor tissue.
Okay, so the stress hormone cracks the gut open, and this mutated super soldier bacteria sneaks out, evades the bloodstream cops, and sets up a base camp inside the tumor.
Right.
But, I mean, a bacteria just sitting there doesn't explain how the tumor grows faster. Is the bacteria actively helping the cancer?
On its own, the bacteria is mostly just an opportunist looking for a safe harbor. But the genomic sequencing revealed the true threat.
The Trojan horse.
Exactly. This mutated egg bacteria was carrying a hidden payload deeply embedded in its own DNA. It was carrying a prophage.
A prophage. Okay, let's unpack that because it's wild. A phage is a virus that specifically infects bacteria, right?
Yes.
But a prophage means the virus has essentially stitched its own genetic code into the bacteria's DNA, and it's just lying dormant.
Like a biological nesting doll.
Right. The bacteria is the Trojan horse, and the viral DNA is the army sleeping inside.
The psychological stress opened the gates. The mutate bacteria provided the transportation, but that dormant virus, which the researchers just named phage-2, is the actual weapon.
So let's do our next quick recap, zooming out on the board for a second.
Okay.
Bullet 1, leaky gut lets the mutated egg bacteria escape.
Yep.
Bullet 2, it travels to and colonizes the tumor using its chemical GPS.
Sure, right.
And bullet 3, it brings along a hidden phage virus. So here's a question for you listening. What if the bacteria inside us are constantly mutating, just waiting for a moment of extreme stress to exploit our weak points?
It's a terrifying thought, because right now the Trojan horse is just sitting there inside the tumor.
Right. What pulls the trigger? What wakes the virus up?
The environment of the tumor itself is the trigger. Yeah, tumors are stressful environments, even at a cellular level. When the egg bacteria establishes itself in that harsh, acidic microenvironment, the stress causes the dormant phage-2 to enter what is known as the ligolytic cycle.
So the virus wakes up?
It wakes up, hijacks the bacteria's internal machinery to furiously copy itself, and eventually just bursts the bacteria wide open from the inside out.
Wow.
The Trojan horse detonates, and massive amounts of foreign viral DNA are suddenly spilled directly into the tumor bed.
See, here is where I would assume the immune system finally wins.
You'd think so.
Right. If a virus detonates inside a tumor, shouldn't that set off massive biological alarms? Shouldn't the immune system rush in, see all this viral DNA, and inadvertently wipe out the tumor while cleaning up the virus?
In healthy tissue, yes, absolutely. But a tumor is a master of hijacking normal cellular functions.
Okay, so what happens?
When the viral DNA spills out, it is intercepted by a specific type of cell called a cancer-associated fibroblast, or CAF.
Now, fibroblasts are usually just the structural builders of the body, right?
Right.
They make collagen, they build scaffolding, but inside a tumor, they get brainwashed. They become these CAFs, forced to act as the tumor's personal support staff.
Exactly. They are enslaved. And these enslaved support cells have a highly sensitive intracellular burglar alarm called TLR9.
TLR9?
TLR9 is a receptor specifically designed to detect unmethylated DNA.
Okay, let's explain that. Human DNA is heavily methylated. It has these little chemical tags on it, right?
Yes, but bacterial and viral DNA lack those tags.
So the TLR9 alarm is basically a scanner looking for untagged foreign DNA. The viral DNA spills out, the CAF scans it, and the TLR9 burglar alarm starts blaring.
But remember, the tumor has rewired the alarm system.
Right. Here's where it gets really interesting.
Instead of calling the immune system for help, the TLR9 activation triggers the CAFs to do something completely unprecedented. The alarm signal travels to the CAF's nucleus and forces it to start expressing a very specific set of genes.
And these are STAR, CYP11A1, and HSD11B1, correct?
Yes. STAR, CYP11A1, and HSD11B1.
Now, I know those sound like random strings of letters to anyone listening, but those three genes are incredibly specific. They aren't immune genes.
Not at all.
They are the blueprints for an enzymatic assembly line.
They are the exact enzymes required to synthesize corticosterone from scratch.
I mean, just let that sink in. The tumor didn't call the cops. It used the alarm to force its enslaved structural workers into building a local, fully functioning stress hormone factory right there on site.
It's brilliant in a dark way.
The tumor no longer has to wait for your brain to feel stressed or your adrenal glands to pump out hormones. It is manufacturing its own private microclimate of pure stress.
It creates a localized, highly concentrated chemical force field. And the researchers demonstrated that this local corticosterone production operates entirely independently of the rest of the body's endocrine system.
This is genuinely mind-blowing, so time for another recap.
Let's do it.
Bullet 1, phage DNA is released when the bacteria bursts.
Yep.
Bullet 2, CAFs detect that DNA using their TLR9 burglar alarm.
Right.
And bullet 3, that alarm forces the CAFs to become local stress hormone factories.
Perfect.
So a question for you. Did you know tumors could literally brainwash your normal support cells into building a localized force field of stress hormones?
Most people have no idea.
Right. Which brings up the ultimate question. Why? Why does the tumor go to these extreme lengths to bathe itself in stress hormones?
Because the tumor is preparing for an attack from your body's heavy artillery.
Okay.
It builds this localized stress force field to paralyze a very specific, incredibly dangerous threat. The germinal center, B cells.
Okay, B cells, we hear a lot about T cells when it comes to fighting cancer.
We do.
They were the infantry doing the hand-to-hand combat. But B cells are like the intelligence and artillery divisions combined.
Yes, and the germinal center is their boot camp. When a tumor forms, these specialized B cells undergo a process called affinity maturation.
Affinity maturation.
You can think of it as a rapid evolutionary sorting process. The B cells are trying to read millions of genetic combinations to craft a highly targeted, high-affinity antibody, specifically an IgG antibody.
Oh, like a custom weapon.
Exactly, one that perfectly matches the unique signature of that specific tumor.
They are trying to forge the exact key to fit the tumor's lock so they can tag it for total destruction by the rest of the immune system.
Right.
But running that biological boot camp, sorting through millions of combinations in a matter of days, that requires a staggering amount of energy.
I mean, it's just this massive metabolic burden. To fuel affinity maturation, B cells cannot rely on standard glycolysis.
Glycolysis is how most cells generate quick, everyday energy, right?
Yeah, glycolysis is like a quick sprint. But affinity maturation is a marathon. So the B cells have to shift their entire metabolism over to something called oxidative phosphorylation, or OXPHOS.
OXPHOS.
Yeah.
That's like throwing pure jet fuel into the cellular engine.
It relies heavily on the mitochondria to churn out deep, sustainable energy. The B cells have to make this metabolic leap to OXPHOS, or they simply cannot produce the anti-tumor antibodies. And this is where the tumor's localized stress factory executes its sabotage. The corticosterone being pumped out by those hijacked fibroblasts binds directly to the glucocorticoid receptor.
Oh, wait. That NR3C1 receptor we saw in the gut?
Yes, the exact same one. It's also located on the outside of the B cells.
The same keyhole, but on a different cell.
When that local stress hormone turns the key in the B cell's receptor, it triggers a catastrophic energy crisis. The biological signal physically blocks the B cell from making the metabolic leap to RxPHOS.
So what does this all mean? The mitochondria get choked out.
Totally choked out.
The B cells are revving their engines, trying to run the boot camp, trying to craft the weapon that will kill the tumor. And the tumor's stress force field just unplugs their power source. They just stall out.
They stall out. The high-affinity IgG antibodies are never produced. And the tumor continues to grow completely unchecked by the immune system. The artillery is silenced.
This sounds almost too perfectly evil to be true. How did the researchers definitively prove that this local stress hormone was the exact thing killing the B cell's energy?
They used one of the most elegant tools in modern biology, genetically engineered mice, specifically a model where they used a genetic scissor to delete the NR3C1 stress hormone receptor, but only on the B cells.
Oh, wow. So they made these MB1TIAN-R3C1LFL mice?
Exactly. Catchy name, right?
Very catchy. So every other cell in the mouse was normal, but the B cells lacked the keyhole.
Right. So the B cells were essentially wearing noise-canceling headphones.
Okay.
The tumor could pump out all the local stress hormone it wanted, but the B cells were completely deaf to it.
And the results?
Absolute confirmation of the circuit. Even when these mice were subjected to the chronic, unpredictable stress model, the energy crisis in the tumor never happened.
Because the B cells couldn't hear the stress hormone.
Exactly. They successfully shifted to OXPHOS metabolism, generated the high-affinity anti-tumor antibodies, and completely halted the stress-induced acceleration of the tumor.
Okay, let's recap this sabotage really quick.
Go ahead.
Bullet 1, local corticosterone hits the B cell receptors.
Yep.
Bullet 2, that blocks the B cells from switching to their OXPHOS jet fuel energy.
Right.
And bullet 3, without energy, antibody production fails and the tumor grows.
That's the chain.
So a question for you. Think about how much energy your brain burns when you are trying to learn a complex new skill, like a difficult language. Your B cells require that same massive, sustained energy surge to learn how to kill a cancer. What happens when you unplug their power source?
They just fail. It profoundly highlights how metabolically demanding an effective immune response is, and how vulnerable it is to chemical signals we usually associate just with our mood.
Which brings me to the elephant in the room.
Mice versus humans.
Exactly. This is a brilliant, beautiful chain of logic. But mice are not humans. We've cured cancer in mice 1,000,000 times over the last 50 years, only to watch those same pathways completely fail in human trials. Did Bashir and her team actually prove this incredibly complex nesting doll circuit happens in human patients?
He did, and the human data is what elevates this from an interesting mouse study to a potential clinical revolution. They analyzed human colorectal tumors and compared them to adjacent healthy tissue from the exact same patients. They found the exact same localized stress hormone factories operating in the tumors.
But what about the bacteria? Human guts don't have the same flora as mice. Did they find that mutated egg bacteria in the human tumors?
They didn't find egg, but they found a different bacterium running the exact same playbook. In the human colorectal tumors, they isolated a strain of Klebsiella pneumonia, which they identified as KPT8B.
So a completely different human gut bug somehow doing the exact same job.
Yes, and this human Klebsiella strain was carrying not one, but 3 different hidden phages. When the researchers tested it in the lab, those specific phages activated the human cancer-associated fibroblasts to produce corticosterone, exactly mirroring the mouse model.
It's an evolutionary loophole that multiple bacteria and viruses have figured out how to exploit. But, I mean, colon cancer makes intuitive sense. It's right next to the gut. What about tumors that are nowhere near the digestive system?
That is where the data gets truly insidious. The researchers reanalyzed massive genomic databases of human brain tumors, specifically gliomas.
Brain tumors?
Yeah. Even in the brain, they found a direct correlation between phage enrichment in the tumor tissue and the activation of that TLR9 burglar alarm.
So this trans-kingdom circuit bacteria escaping the gut, detonating viruses and triggering local stress receptors, is a universal blueprint. It's a mechanism that multiple types of human tumors in completely different parts of the body can use to shield themselves.
Which means it is also a universal target for therapy. If tumors are relying on this elaborate biological Rube Goldberg machine to survive, we can break the machine. The researchers tested 2 different clinical interventions in the lab to close this loophole.
Okay, let's talk about the counterattack. How do we break it?
The first intervention was a blunt instrument. If the entire cascade requires a bacterial Trojan horse to deliver the viral payload, you just eliminate the horse. They injected a common antibiotic, ampicillin, directly into the tumor.
Okay, no bacteria means no virus. No virus means the burglar alarm never goes off and the local stress factory is never built.
Exactly. But they also developed a precision instrument. They injected a specialized drug called ODN-2088. This compound is a TLR9 antagonist.
An antagonist meaning it blocks the receptor. It's essentially a pair of molecular wire cutters for the burglar alarm.
Precisely. With ODN-2088, the bacteria can embed in the tumor, the environment can trigger the lytic cycle, and the viral DNA can spill everywhere. But because the TLR9 alarm is cut, the CAFs are deaf to the signal. They never receive the command to start manufacturing the stress hormone.
And did both of these interventions actually rescue the immune system?
Yes. In both scenarios, whether they used the antibiotic to kill the bacteria or the antagonist to cut the alarm wire, they successfully stopped the localized stress production. The B cell's energy grid came back online, they switched to OXPHOS, and the immune system began fighting the tumor again, despite the ongoing systemic stress the host was experiencing.
That is a staggering clinical insight.
Yeah.
Let's do one final recap of the human side.
Okay.
Bullet 1. Human tumors use the exact same circuit with different bugs like Klebsiella pneumonia.
Right.
Bullet 2, it works in colorectal cancer and even brain tumors like gliomas.
Yep.
And bullet 3, antibiotics or TLR9 blockers like ODN-2088 can reverse it and save the B cells.
That's the breakthrough.
So here's a question for you. If you were a doctor, would you have ever guessed that prescribing an antibiotic could actually help the immune system fight a tumor during times of extreme stress?
It forces us to completely reevaluate the tumor microenvironment. This raises an important question. Could the future of cancer therapy involve treating hidden bacterial and viral infections that are actively colluding with the cancer?
Or deploying localized drugs to block the specific stress receptors?
Right.
It is time to step back and look at the whole board we've uncovered today. We started with a feeling, the heavy, abstract blanket of chronic, unpredictable psychological stress. And Bashir's team showed us exactly how that feeling becomes physical.
It's quite the journey.
Systemic stress hormones crack the mortar of your intestinal walls. A highly mutated bacterium slips through that breach, navigates the police bloodstream using a chemical GPS, and colonizes the hostile environment of a tumor. Once there, it detonates a hidden virus. That viral DNA trips an alarm in the tumor's enslaved support cells, tricking them into building a local stress hormone factory. And that local stress hormone stars your B cells of the mitochondrial jet fuel they need to forge tumor-killing antibodies.
It is a continuous, unbroken, trans-kingdom chain. Psychological stress to gut leak to bacteria to virus to local hormones to immune failure.
Wow.
But understanding the specific mechanics of that chain is profoundly empowering. It takes the vague terror of stress and transforms it into a concrete biological pathway with multiple vulnerabilities that we can target with medicine.
We have this deeply ingrained habit of treating our minds and our bodies as if they operate on completely different frequencies, as if a tough week at work is somehow fundamentally separate from the cellular biology of our immune system.
But this deep dive proves that the wall between mind and body just doesn't exist.
It really doesn't. Which leaves us with a final provocative thought to mull over. If our psychological stress can trigger our own gut bacteria to mutate, escape, and release viruses that ultimately decide the fate of a tumor, does that mean our microbiome acts as a literal physical bridge between our emotional state and our cellular reality?
And if our bacteria are reacting this violently to our stress, what other thoughts, what other emotional states are they listening to right now?
The communication goes both ways, and we're only just beginning to learn the language. Thanks for taking this deep dive with us today. Keep questioning the things you think you know, and we'll see you next time.